Happy Earth Day, everyone. It's an interesting one, to say the least. Let me share. I'm going to go through three narratorials, is what we call them, tutorial narratives. And I hope most of you are somewhat familiar with KBase and done one of the intro webinars, because I'm afraid I don't have time to go through the basics of things. So let's see, Ben, or can you see a narrative now?
Yeah, it looks good. Everything looks big enough, at least on my screen. Okay,
fantastic. So I'm not going to be able to go through all of the basics of how you get to this point, unfortunately. If people want to follow along, other than looking at this shared browser and are logged into KBase, you'll be able to see this narrative. It's number 22290, where you can search for Build a Gene Tree tutorial. So that's the first one I'm going to go through. Fortunately, I'll go through a little bit of the foundations of using KBase in the narrative. So for example, the layout of a narrative, this is one analysis session in essence. It's it's like an e-notebook kind of, but it can be computed. And in the upper left here, I'm hoping you can see my cursor moving. This is the data pane, and in it we have different data objects, and data is represented in KBase as objects that are structured, and so you can examine information like this tree is a structured object, and you can get information about it. Down here is the app pane, and you can search for apps in it, like build species, well, that's the wrong one. Microbial species tree. All right, I'm doing a terrible job here with searching. Brass, no? Okay. Let me find one that actually has a name. There, Annotate. Ta-da! So you can do Annotate and look at the different apps that match that and so on, or you can just dive into these different categories. And to launch an app, you would just click on that and it opens an app cell in this notebook panel. So let me delete this. Okay, so that's how we wind up with these notebooks. There's data objects and there's apps. And so here is an abstraction of the sequence of steps in this narrative notebook, where a data object I've got represented as a circle and an app as a rounded square. And so you can sort of imagine that analysis steps are, these app runs are sort of taking some data and turning it into a different type of data, and it's connected, and then outputting visualizations and so on. But it's a lot of object type, data type transformations in a sense. So I'm going to hide the data and app panes now, so that we have a little more real estate to look at the notebook narrative. And so these steps we're going to go through here are just sort of to get you warmed up with using apps and to illustrate sort of a granular analysis that is kind of akin to the steps you might do on a command line. But all of this is going to be done using a graphical user interface instead of a command line to make it easier. Additionally, all of the compute was performed on our KBase cloud in the background. So what you're seeing is here is going to be the result of running each of these steps. I'm afraid I don't have time to run these apps themselves. I believe this is only an hour and a half, so I may wind up going somewhat quickly as a consequence, and please ask questions in the Google Doc. And I won't have time personally, but there are other KBase personnel ready to answer questions in there, and I will go back after this and answer questions as well. Okay, so gene trees are one of the sort of foundational elements of phylogenomics, where you want to understand the evolutionary relationship of a gene family and the genes that are present within each species. And so we're going to go through and we're going to find, in this case, all the sulfate adenylyl transferase genes in a set of related organisms, and build an evolutionary tree from them. And I happen to know this one's an interesting case because I did the genome analysis on this organism, Candidatus desulphorutus adaxviator. So we're going to start with a sulfate adenylyl transfer. Well, no, we're going to start with the genome. And so this genome is in KBase in our RefSeq reference database. And we're going to run this app here where we insert genome into species tree. And so one of the steps I've skipped here is going and pulling in the initial genome object. So if I was doing an intro to KBase, I would go here and I would go to public and I would have these NCBI reference genomes and I'd search for desulphorutus. And I would have it here, and then I would add it to my workspace. I'll just do it. I hope that doesn't mess things up. Okay, so I've added it, and so that's how we got the genome in from RefSeq. And this version, it's a little bit old, and when we imported it, it wound up having an object name GCF, which is the NCBI handle for it. So we start by taking this and going and picking this genome as our starting point, and we're going to pull in, say, 20 RefSeq relatives. And this is important for this type of analysis where we want to copy the genomes to our local workspace. So these reference genomes in the RefSeq database, our copy of the RefSeq database, are in a different area of KBase. They're not this narrative. And so for a lot of these apps to be able to see these genome objects easily and analyze them, it's beneficial to have them in the local narrative. And so that this copy public genomes to your workspace will pull them and make local copies, and it'll make the species tree that's generated refer to those local copies and not the remote RefSeq database. And so we're going to have two output objects from this. We're going to have this output tree object, and we're going to have a genome set that corresponds to that. So it's going to create a species tree. It's going to pull these genomes that are in that tree from the RefSeq database and put them into our local narrative. And it's going to make a genome set of those local copies. And we're going to be able to operate on that genome set and on that tree later, the species tree later. So when we run that, you can see up here there are 3 tabs. This is the configuration tab, which is what your first, when you launch a new app, comes up. This is the job status, where we get log information about its run and a lot of information about how it's doing. And then this is the results tab, where let me just close this again, where we get our tree. And in this case, the result gives us this HTML report embedded in here, which is an image. And we can open that up into a separate window if we need to see it bigger. We can play around with its sizing. Oops. Its sizing if we need to. Additionally, there are links. So this is the HTML. We can download that, and that'll have that image embedded. And then there are files that are generated if you need those. So in this case, we're getting a Newick version of this tree that has sort of KBase IDs in it. There's one labels where the IDs have been replaced by these names at the leaves. PNG version of it, which is what's shown here, or if you need a vector version, there's the PDF. And so lots of these apps are going to have extra files like that. I'm not going to have time to detail all of it, so I recommend you look at them if they're file types of interest to you. So that's on top of the objects that are generated. Now, the objects that are generated are the things we can use in KBase in subsequent steps. These are more just for you to use in your publications, or if you have a special tool that doesn't happen to be in KBase you want to look at the data with. Okay, so we're going to take this, and as you can see, we've got our 20 relatives in here, but some of them are a bit redundant. We maybe don't want so many of these because we're going to be doing, more analysis, and so we might want to enforce a little bit more phylogenetic diversity and not sort of waste compute cycles and on what are pretty close relatives. So we're going to remove the redundant genomes from the species tree. So that's this next step here. And so to do that, we can use this remove genomes from Genome Set app, and we can go and we can specify these local copies. These will be ones you can pull up because we made local copies of them. And we're going to remove them from this genome set we generated, this -all genome set with all 20 of them. And then we're going to make a neighbor's genome set that's smaller. And here is, if you have non-local genomes, you can put the name, here. If you have non-local genomes, you can add them with the object name, because they won't show up when you try and configure this part. You have to do that manually. So that's important if you didn't copy over the genomes and you're still using the refseq versions that are non-local. Sorry if that was confusing. Okay, so we're going to remove that. In this case, the result's not all that fancy. And so we're going to wind up with just a report of how many are left in it. And we can look at one of, now we're going to want to find the sulfate adenylyl transferase gene. And we're going to do that by launching this object, this genome object, and putting it into the narrative notebook frame by just clicking on the name of that object, and it'll launch it here. So this is duplicative, but so now you can go and you can browse the features, you can search for sulfate, you get all these sulfate genes. Maybe we want to be more specific, like that. And so now we've got some, both the CDS version and the gene version, and lo and behold, we have two. So now we have the question, are these two copies the result of a pyrologous expansion within the desulfurudis clade, or the immediate lineage, or is it an earlier duplication of this? Or was it a horizontal transfer? Or we don't know the origin of this, but the question is it more recent or earlier on? And so we're going to go ahead, and that's the question we're going to try and answer with this gene tree. And so we're going to take one of them, just arbitrarily picked one of them, and we're going to do a BLAST-P search, and we're going to use the genome set we generated, and we're going to paste in the sequence in the sort of traditional BLAST way, web BLAST way. And here, because we're putting the sequence directly in, we don't need to put in a query object. It's sort of one or the other. once we've run BLAST, or if we had a query object previously generated of just a single gene sequence, then we would input that here instead, and I'll show you that in a minute. But for right now, we're just starting out with a sequence. It could be any sequence, but and we're going to go and we're going to search, and we're going to use the default sequence identity of 40. We're not going to get in, this advanced options. We're not going to change any of the advanced options like alignment, overlap, threshold, or max accepts. You can have additional BLAST format output. We're going to stick to this format 7, which used to be format 8 for old folks. And that's going to be our extra text output. But we could have had the pairwise version or whatever. So we're going to run that, and the results, we're going to take a look at the results, and we're going to see the results of our hits. And this time we're getting a created object that's a feature set. And so this is all of the genes that are hit by the query in these genomes that are in this genome set. And so here we go, and we get 100% identity to our query, yay. And then, but if we scroll down, we see that there are some of these marked in gray that are not winding up in the feature set. So they're still hit by the BLAST search, but our thresholds for inclusion in the feature set were tighter than would be necessary to include these genes in the results of the hit. So you can pick your thresholds by coverage, and maybe you don't want short hits to count, or sequence identity or e-value or whatever. And so in red here is the reason why these were chucked. And so if you were going to redo the search to include these in the feature set, we'll need to drop the sequence identity threshold below 29.2. And so that's what we're going to do in this next run. We're going to rerun BLAST, and we're going to drop the sequence identity threshold to 20 and check that these are indeed winding up in this 20% identity feature set. And yes, indeed, they all made it in. Okay, so now we have a feature set of sequence homologs. And so we're going to build a sequence, a multiple sequence alignment of those. The typical steps in phylogenetic reconstruction is you build a multiple sequence alignment, you trim it for variable regions if the sequences that are present in it are normal distance or greater, because there are often these variable regions that are less phylogenetically informative. They may correspond to loops in a protein or something like that, which are more rapidly diverging than the core scaffold of that protein. Or RNA or something, ribosomal RNA. So in this case, these are the kind that we're going to want to go and build a multiple sequence alignment. We're going to do it with muscle here. There are a bunch of advanced, well, I guess there aren't a bunch of advanced parameters. You can decide how long you want it to run before it gives up. If it's a big one, you want to increase these values. And so we go and we run it, and we get the multiple sequence alignment shown here in Clustal. And you can download it as FASTA or Clustal, but it's going to generate an MSA object, which can be seen here, the different MSA objects. Okay, so now we have our multiple sequence alignment object here, but like I said, there's some more variable regions that are less reliable for computing phylogeny, and so we're going to trim those out using, in this case, G-blocks, tried and true method from yesteryear. And so we're going to go and we're going to run it, and we can see that those gappy regions have been removed. And so, hooray, now we have something that we can compute a tree from. And so now we're going to use, in this case, FastTree. And this is a tool from the Arkin lab, which we like because it runs very quickly and does a very good job computing reasonable topologies and Bootstrap scores that are pretty meaningful, reliable, but it's quick. And so here we go and we take our input MSA, the trimmed one, and we're going to generate this tree. And it's not a species tree, so we're going to uncheck this. And then there are quite a few parameters. So now if you are a command line type of person and you're using KBase, because of how it integrates all the tools and the data, and it has value for that reason, you can still, in some of these tools, really get at a lot of the command line options and play around with them if you so choose. But otherwise, you can just stick with the defaults that are not in advanced. And so now we have our gene tree. And let's look at it. So what was our question? Our question was, are the 2 copies of sulfate adenylyl transferase into sulfurudis recent paralogous duplications, or is it more ancestral, or horizontal transfer? So we can see that they are not grouping as sister in this tree. There's one here and there's one up here. And so that suggests it's either an ancestral duplication or horizontal transfer. But since not everything, if we go and we look at these, there are not, everything at this lineage and below does not have 2 copies paired along here, that suggests it is a horizontal transfer and not an ancestral duplication. Okay, so we just learned something using the gene tree. This is a very good way to use this tooling if you're just doing it for one homolog collection. If you're trying to do it for lots and lots, it's a bit laborious to go through all these steps one by one, even if it gives you that precise control and allows you to look at things like more carefully and play around with advanced parameters. But if you want to do it a lot of times, I have more recently, because it was just such an obvious thing to do, taken those above steps and linked them into this build gene tree app. So you can just run it. Once you have a feature set, you still have to get the feature set by some mechanism, be it a hammer search or a blast search or some gene family annotation. And so here we're going to do the same thing. We're going to start with a 20%. However, in this case, because we're chaining a bunch of apps together into this one sort of Uber app, I guess that name's taken, larger app, we break out and put the different parameters for each of the stages in as advanced parameters. So muscle parameters, G-blocks parameters, fast sheet parameters in advanced. And the results, again, it's pretty much the same. Well, it is the same, but we wind up with lots and lots of stuff available from this one app. Okay, so that's the build a gene tree tutorial. And I think I'm pretty much on time, so that's good. So the next one is a bit more involved. So here we're going to start getting at more whole genome analyses. So we're going to wind up taking a bunch of related species. In this case, we're looking at a subfamily of largely sulfate-reducing Clostridia. So Desulfurudis edax Viador is one of them. Its closest relative was obtained by my colleague Sean Youngbooth. It's called Desulfurudis cowani, and it was done by direct environmental sequencing and genome extraction of, metagenome assembled genomes, or as they're called these days, MAGs. So this is a a MAG is the input, which technically desulfurudis is kind of sort of desulfopertinax. And so we're going to do that same thing where in this case, we're going to start by uploading just the assembly of desulfopertinax. We don't have the gene calls yet. And we're going to go through these steps where we take it, we annotate it, we put it in the species tree. We're going to get a genome set of related genomes. We're going to trim that down. And then we're going to do annotation, functional annotation of that genome set. And we're going to get canonical gene family annotations, and we're going to be able to slice different functional subsets from that genome set. And then we're going to be able to look at combinations of the species tree and the domain annotation to look at phylogenetic conservation of function. And then I'll talk a little bit about gene family profiling that is sort of targeted instead of this whole database scanning. Okay, and then after that we have a pangenome part, which is in part 2. Pangenomes can be quite large, and so I broke it up into a separate narrative to help us focus. So we're going to go through, we're going to upload desulfopertinax cowni. So again, here what it is you go and you click on the import tab, and you can pick one of these, and you can select a format. And in this case, it would be an assembly here. And then you click on this, and it would launch an app cell for import, which is where this guy came from. And so here we have the desulfopertinax cowni assembly, and you had to specify the FASTA file from your staging area, and it's a MAG, so we picked the type of genome. We can threshold the minimum contig length, and I really emphasize throughout analysis, you probably, unless you're doing something very specific with small RNAs or something, for the most part, you want contigs that would fit a protein. If proteins are what you're sort of trying to base your functional analysis on. And so 500 is just on the edge of a normal protein size of a smaller one. So this helps us remove the sort of long tail of short contigs that. And so, all right, so we have this assembly, and this represents the contig sequences, but that's it, no gene calls. And here we have it, and we can browse the contigs, and here they are, and here are their lengths, and so on. And so we've got 6. So, but we still need gene calls. So we're going to take desulfopertinax assembly, and we're going to annotate it. And so we're going to use, in this case, RAST. We also offer PROCA. And we're going to take the assembly, and it's a bacterium. So it has genetic code 11. And we're going to create a RAST genome object. So this will be the assembly sequence and the gene calls and RAST functional annotations, many of which come from the seed annotation namespace. And so the result is this genome object. That's a KBase genome object that we can then use in subsequent stages of analysis. We can browse features, as I showed before, and so on. So does it have sulfate adenylyl transferase? I don't know why I picked such a long word to type. So here, oh, it even has 2. So lo and behold, if we, oh no, sorry, it's just 1. It's the gene and the CDS. Okay, so that would suggest that the desulfurudis sulfate adenylyl transferase, the duplication, the extra copy arrived post split from desulfopertinax. We'd probably want a few more genomes to make that statement, and of 2 is not that reliable. But it at least suggests the possibility. Okay, so we're going to take we're going to find the near relatives using the species tree. We're going to insert genome into species tree. Here we have the desulfopertinax as our bait genome. We're going to go, we're going for 20 here, and we're going to try and get the proximal ref-seqs. And here's our tree. So now we're going to remove the redundant lineages, these desulfotomaculum. And these are the ones we choose to remove. In this case, since I was working in a way that allowed me to really dig into this, instead of just arbitrarily picking one or the other, I actually went and looked at the genomes themselves and checked to see how many contigs they were in, and with how many gene calls they had and so on. So I used some sort of expert logic and close attention to decide which of the two to chuck. So here I've gone and done that, and I'm getting rid of 5, and I'm producing this non-redundant genome set here. And now I want to annotate them. So I'm not going to change the gene calls themselves, but because this is straight from RefSeq, it doesn't have the RAST annotations that a lot of our tooling likes to use in its ontologies. So we're going to go and annotate all of these with RAST, just to make sure they're all in that foundational namespace and consistent in KBase. And so here we're going to go and we're going to use this Annotate Multiple Microbial Genomes app, where we can feed it a genome set to produce a collection of RAST annotated genome objects. And this is, again, giving us the functional annotation, but not changing gene coordinates at all. And so we go and we do that, and we get our annotated genome objects, these .rasts. Now we're going to use that and build a new species tree using the trimmed genome set with the RAST annotated versions of those genome objects. And so that was one of the outputs of this multiple microbial genome annotation, is this RAST version genome set. Okay, so we're going to build a new species tree using that. Now, in this case, we've already pulled in our RefSeq proximals. So now we want to just build a tree with our local copies. And so there's this Build Microbial Species Tree app that can accomplish that. So we're not going to get any more RefSeqs here. And one of the nice things about this guy is that you can also add in a phylogenetic skeleton. So we have the possibility of adding in bacteria and archaea. So there's one phylum exemplar that we can sort of add in. And so we could add in just bacteria, isolates, or so on. In this case, the application we're looking for is really more local functional conservation and profiling. And so it doesn't make sense to add in very distal phyla. But if you have a collection of genomes and you're not sure where they fall, there are a couple of options. One is this option, where you're going to build a tree with these phylum exemplars as the skeleton. Another is we have GTDBTK, which is a method from Phil Hugenholtz and colleagues, Donovan Parks and so on, that is very good at classification. And so you could use that also. Okay, but in this case, we're going to stay local to this clade. And so we're going to come out and we're going to get a species tree for these guys. And that's shown here. One other thing I should mention, you can see these are all in yellow. Another thing you can do is, if you have additional genomes you want to add beyond the initial query set, you can add those in here, and you can pick which colors whether you want to color them or not. Okay. So we've done our RAST functional annotations on this set, but we additionally want to go and annotate with COG, PFAM, TigerFAMs. Maybe we want to just have a few different namespaces or more comfortable in one of those for functional classification. And so this annotate domains in a genome set is an option to run that. Now, it runs them currently. It runs them one at a time in serial. If you want to do it in parallel at this moment, I'm hoping to fix that soon, but I haven't yet, or KBase has not yet. We have to do it in serial, or alternatively, you could launch annotate domains one at a time. If you have a small enough number of genomes, you can run them in parallel, but you just wind up with one app cell per run, and so that's the cost. In this case, I was willing to spend the time and just wanted the cleaner real estate, so I just ran it there. Okay, so now we're going to start looking at functional profiles for these things. The view functional profile has sort of three versions of it, but it's the same thing essentially. So there's one for genomes, there's one for species trees, if you have that, and there's one for feature sets. And I'll talk first about the sort of base one where it's a genome set that you're using. So we have the ability to do just sort of a top level scan. In this case, we can just do sort of the COG functional categories, and we're going to do tallies of how many genes are present in these genomes, and it's going to be represented in this case as a percentage of the annotated protein coding genes, meaning annotated by COG in this case. And there's some options. You can choose how you want it to display when it's output. You can choose heat map or just the raw numbers. And I won't go into the custom domains yet, because that's if you're starting to drill down on functions. So you can imagine this as sort of a top level summation of some function if you want to see if there's anything particularly weird about it. And for the most part, that's not the case. So these top level functions, cell cycle control, cell division, chromosome partitioning, yes, they all have it. So that's good. If it was particularly, if these are, for the most part, pretty complete genomes or isolate genomes, and therefore that's not too surprising. But this would be a useful tool, especially if you have a MAG that's somewhat incomplete and you want to get a sense of how complete it is in that area. So that's sort of the top level COG function. You could do the same thing, just reporting the numbers. In this case, suppose we were interested particularly in COG class C, or functional category C, energy production and conversion. Then we could go and we could run, instead of using the top level COG, we could use custom domains. And if you click on the option here to do that, you'll wind up being able to either explicitly name a COG or other gene family explicitly, or we can go for the subcategory C, and it's going to expand out the hits. In this case, I'm showing the raw count instead of the heat map. And so if you go and look at this, this is COG C. So these are all the COGs that are in COG C, right? 1301, sodium proton dicarboxylates importer, and so on. Anyway, so we get an exact tally of how many genes are hit by that COG in each of these genomes. Now, this is just the tally, but suppose we want to actually get those features from this tool. So we want them to be output. Well, to do that, it's too many if we use these sort of top level categories or even these mid-level expansions of a tiger role in this case, or a PFAM clan in this case. Now, if we explicitly want the genes in a feature set for COG 578 or this tiger fam 2034, maybe I should make this bigger. Then what we would do is when we're doing the query, we're going to use custom domains. We're going to explicitly request COG 578, PFAM 7, Tiger 2034. And when you explicitly request a particular gene family, then it will create a feature set for you. So you'll have those genes in hand to build a gene tree or whatever. But we're still, suppose we want to expand, more generally, and we can do that too. And so we can sort of mix together, if we so choose, this type of analysis. So here you can see we got these feature sets. There turned out there was no PFAM 7, so I didn't make one, or no hits to PFAM 7. Okay, so you can see we've got a collection of different things, and so you can customize this however you want. And you'll see in the key, we're only, in this case, showing those gene families which had a hit. And so grayed out are those with no gene hits. Okay, so we can look at COG 578 by clicking on the feature set over here to drop it into the notebook. And so we've got these different features, and we can browse them and so on. Additionally, we can put these into phylogenomic or phylogenetic context, I should say, by combining a species tree with the annotation. Now, these domain annotations, there's lots of them, and they're auto-detected for each genome in your narrative. This particular app goes and finds the domain annotation that corresponds to a genome, because there's a link in the domain annotation. So this app will scan it for you. You don't manually input them. So we're going to, in this case, we're going to take the species tree. The app will go into the species tree, find the genome objects referred to, look in the workspace for, well, in this case, I'm using RAST annotation. So it's going to just take it from that genome object. If I was using COG or PFAM or Tiger fam, then it would look for a domain annotation object. And it'll go and it'll put this phylogenetic functional profile together, where we've got the different, in this case, seed subsystems listed along here, and but it'll be against the species trees. So rows are, in essence, wind up being ordered by species tree instead of alphabetically or some other way. And now you can see what all of the members of these classes were. So sporulation is present, and so on. Sulfate reduction, etc. So next we have so that's sort of general whole genome, whole namespace annotation. But we may want to go and do something more targeted. We may be interested in particular functional classification or trophic assignment, and don't want to spend all the time using all of COG or all of PFAM. And we may just have a custom collection that we want to use. And so this is sort of one example of a collection of gene families that will work for doing on-the-fly assessment and visualization of gene hits using the KZ family. This is dbCAN collection of hidden Markov models. So if you're interested in adding another collection, please come talk to us. There's a contact us link that you can use to reach us, and we can help you use this as a template to build your own collection. So we're going to go and we're going to take, in this case, KZ, and we take this genome set, and we can subset whether or not we want any of these subsets of KZ, GHs, GTs. If we leave the defaults and don't add anything specific, it will scan all of them. And you can, it creates feature sets. You can do a separate object, feature set object for each model, which winds up making quite a lot, or in this case, we're choosing to merge the hits within a KZ category. So we're going to get all the GHs and so on. Now we can subsequently slice feature sets by genome. There are a bunch of utilities for manipulation of genome sets and feature sets that allow you to pull out by genome or other ways the subset of features. Okay, so we're gonna use this as the sort of base name. So it'll be GH, GT, and then this base name. And so we go and very similar to view functional profile, we'll get a heat map. And this will reveal, how many copies of this there are, 2 in this case, all scaled by maximum value. And we can actually go and look at the results of the hammer search in a table very similar to the BLAST. And so you can see where it's maybe not getting the values on those thresholds. So, okay, so you can dive into it a little more that way if you so choose. Now you may also have gene families or collections of sequences that aren't necessarily part of COG yet, or even some collection of hidden Markov models that was already produced, and you may have these sequences from your own studies that and your own genomes or your own sequences, and you can take those and you can build multiple sequence alignments of them, and then use that to query your genomes using Hammer. And so if you have MSAs, you put them into your narrative, or you build them in your narrative. And if you you can either pick that subset of MSA objects that you want to use as the queries, or if you leave it blank, again, it'll detect it in your narrative notebook and use any MSA that's in this particular narrative. And it'll go and it'll scan them, scan your genomes, and report back. It'll make feature sets from each of those genomes against that model, like CDHA, DNAA. Well, no one cares about DNAA for this purpose, but and so you'll get a tally of how many hits there are to your custom MSA query. And this is nice if you're in the middle of your research and it's not a published collection of genes, and so this can all be done without in a private narrative on your own data. Okay, so that's it for the comparative functional assignment. And I'm going to move on to the next tutorial, which is number 29419 here. And so here we're going to start talking a little bit about the pangenome view of a collection of related species. So a pangenome is it represents the full genetic complement of a collection of related species. So if you were to take all of the genes in all of these species and put them into a big bag, that's everything that's present in that collection of genomes. So as evolution happens, genes are acquired, lost, evolved, and you can have paralogous duplication and evolution off of that. And so there are a variety of avenues to functionality gain and loss. And so pangenome analysis is very useful for determining what's essential for a lineage and what's changing around or particular to some branch of that lineage or even just one species within that lineage. And so there are terms like core pangenome, where all or almost all of the genomes in that collection have a gene with that functional classification, or in this case, we're doing pangenome analysis using sequence homology, which is tells you whether or not they're evolutionarily related genes, even if they have, anyway, functional annotation's a whole another can of worms. So we're going to use sequence homology to determine evolutionarily related genes, and then we're going to take that and we're going to figure out, across this collection of related genomes, how much that sequence cluster functional homologs are present or absent, or if they're in sub-branches and so on. So that's what we're trying to do here, is we're going to try and put a phylogenetic context to pangenome analysis. This isn't necessary if you have a bunch of very closely related strains where everything's really tight and you're just asking, more recent gene gain or loss, or even just sequence variation. This is more for longer term genome evolution. So in this case, we've got our sulfate reducing Clostridia, and these are indeed a perfect candidate for this type of phylogenetic pangenome analysis. And so we're going to take our collection of sulfate reducing Clostridia. In this case, I've got a different name for the genome set. It's Clostridia, and I'm going to add in an outgroup, because this type of analysis where you want to ask if something's core, that means it's present in everything, but we want to, for this type of analysis, we're not necessarily interested in DNA A, as I mentioned. It's, it's universal, it's housekeeping, we know about it. So we're going to introduce E. coli, MG1655, as our outgroup, because we know that that's distant in evolutionary space, and so we can use that to identify housekeeping. So if it's present in E. coli and all these Clostridia, then it's housekeeping. If it's present in all these Clostridia but not in E. coli, then it's clade specific. Okay, so we're going to add in E. coli to our genome set, and we're going to operate on that genome set. And so you can take a look at the genome set, who's in it. Yes, our E. coli was added here. And then we have a bunch of these Clostridia. And now we're going to build a pangenome. And this pangenome calculation is going to take all of the gene sequences. In this case, we're going to use the genome set. We could add them in one by one with these genomes if we so chose. And then there are a bunch of advanced parameters for ortho-MCL. And that's going to produce this pangenome object. We've put in 17 genomes, so it's the 16 Clostridia plus E. coli, and we're getting out these ortholog groups. And you can take a look at the pangenome and look at how many genes there are in each genome, how many of those wind up in homolog clusters, how many are unique to this genome, and homolog families. There may be more than one gene in a homolog in the same homolog cluster, and so that would be this concept of homolog families. So we get a sort of summary of that clustering. You can also do pairwise comparisons across them to see, of this alcohol of borax against acid oxidants, what the overlap is in their genes. We can go and we can look at the families of homolog clusters, and it takes a little while to load because these are large objects. Anyway, so there'll be a family ID, a protein coding gene count, and how many genomes it's found in, and so on. So here, for example, this cluster, there's 62 genes that are in this cluster from the 17 genomes, and you can see how many genomes it's found in. Okay, so this is nice summary information. It gets us started. But now we may want to ask things like conservation in a more whole genome way. And so there are a couple of ways to do this. One is we can build a circle plot of the pangenome. Now in this case, we need we need to pick one of the genomes to use for the coordinates. Okay, so in this case, I'm going to pick Desulforudis adaxbiodor as the base genome, and I'm going to take the pangenome, and I can pick an outgroup, or I don't have to use all the genomes that are in the pangenome, and so you can custom compare genomes here if you chose to. If you don't pick it, then it will use all of them. And the outgroup genomes, you can specify which member of the pangenome, in this case E. coli is the outgroup. For some reason that has been lost in the display here, but that's what I did. I'll have to fix that. So here we've got, you know what, this is hard to see in this. So I'm going to view it in a separate window and shrink it down a bit. And we can take a look at the circle plot of the pangenome. And you can see here on the outer ring, we have the Desulforudis adaxbiodor genome. And then aligned by homolog group are in each of these inner rings is one of the genomes in the pangenome. And the color indicates whether or not the classification of each of those genes in the homolog cluster. So for example, and if there's nothing, if there's no match to the homolog cluster, then it's a gap. Okay. So you can see there's gaps here. So these are sort of more variable regions. Now we've got different colors on the outer ring from the inner rings. And that's largely to reduce eye strain. But so for example, in light blue here are the in between pangenome classified as non-core genes. In red are the singletons that are only found in the Desulforudis adaxbiodor and not in the other genomes. The core plus outgroup, meaning including the E. coli, are in dark blue, and the clade-specific core, so found in all of these guys, but not E. coli, are in magenta. In the inner rings, we use slightly different color because again, this blue would be too bright, and so the gray is that in between singleton and core. So it's nice to visualize and get a sense of which regions are sort of evolving more rapidly or more different from, contain genes that are more different from the rest of the lineage. But additionally, we want to slice out some of those functions. So here we're going to go and so that pangenome plot additionally has the capability of producing the clade-specific core, the universal core, the singleton set, and the in-between, the variable pangenome. And we can take that and use it as input to our view function profile. But in this case, it's sliced out feature sets. And so we're going to take that feature set and do a functional assessment of this clade-specific core for the Clostridia. In this case, we're going to use the seed rolls as our starting point, and then we could drill down as we wish. But in this case, we're going to start with the clade-specific core. And so these are the genes that are gene families, I should say, that are found in the Clostridia and not in E. coli. And so we've got all of these. Now, again, this is not clustered by functional annotation. It's clustered by sequence homology. And so, you know, a lot of this stuff, it may be a bit distant from E. coli, and therefore some of these may be present in E. coli. It just was distant enough that OrthoMCL did not recognize that sequence homology relationship. So again, play around with the parameters in the OrthoMCL clustering to make sure that you're getting the results you're looking for. Sorry, the quality of results you're looking for, not cherry picking. Okay, so you can do the same thing for the non-specific core or the flexible pangenome, and get those out. And again, you could use, you know, specific, if you were to zoom in on alanine biosynthesis and then pick a particular explicit gene family, it'll slice out just that feature set. And then you can build gene trees or whatever. Singletons, much more sparse, obviously. And so, anyway, that's the idea of doing the functional assessment of those. So next, we're going to take this and put it into a phylogenetic context. So there's these pangenome accumulation views of pangenomes, where as you pick one genome to start, and then you add in one by one and recalculate the pangenome core, singleton, and flexible portion, and you'll see the core goes down. I guess I should go this way. The core goes down, the flexible goes up, and the singleton goes up. But maybe that order that you're adding them in is somewhat arbitrary, or it doesn't take the phylogeny into account. In this case, we have distantly enough related lineages that we can build a species tree where the pangenome calculation becomes meaningful. And so here we're going to use this phylogenetic pangenome accumulation view of it, where we take the pangenome object and the species tree, and we combine that information into a phylogenetic view of that pangenome accumulation. In this case, we can slice out the feature sets for core, singleton, and flexible as separate feature sets. And in this case, we're going to do it for every single node in this tree. So it can create a fairly large number of objects, and you maybe want to run this without doing that the first time. So here we have, again, I'm going to expand this into a separate window. And so we'll go through, we'll have the species tree, and we're going to have nodes up the species tree, and we're just arbitrarily numbering them 0, 1, 2, 3, and so on. And here we have a tally of how many genes fall into the singleton, partial, or perfect core for each of these nodes. And there'll be a feature set associated with each of these bars, not the total bar, but the others, for each of these nodes. And here is a pie view of them. So as you can see, in blue is the flexible pangenome, in red is the singleton. The notion of core doesn't apply to 2, because it's either singleton. Oh, sorry, there's no flexible. There's only core or singleton, because the notion of flexible doesn't apply here. And as you can see, the red singleton goes up as you go up the tree, partial goes up as you go up the tree, and core goes down. So now we can ask questions about, for example, what is sort of the clade specific core for this branch, starting from node 12 below. So here we can imagine that this acid oxidants becomes the outgroup to these four guys, and we can go and do some tricks where we take the feature sets and slice them with a Venn diagram type slice. So we can take feature set A, like I mentioned, the core pangenome for node 12. That means it's found in all four of those, oops, wrong one. It's found in all four of these lineages. And then the non-core, but also not singleton, the flexible fraction of the pangenome for node 11. And so that's here. So let's think about this. So if it's universal for these four and not universal for all five of these, then that means whatever genes are in that intersection are the clade specific core. So if it's flexible, that means that this is the only one where it's absent from, right? So we can do that Venn slice. And so the intersection features found in both A and B, and we'll call that node 12 clade specific genes. And that'll make that feature set here. And now we've got that feature set, and we can do a functional assessment of it. In this case, arbitrarily, for illustration, I picked TigerFam, some different TigerFam categories. And we get that tally for these four guys in that branch of the tree, okay? So this is the clade-specific core for this branch here. So you can ask questions along those lines. So that's actually all I had prepared for now, which I guess is good. We went pretty quickly. I don't know if people, I don't know, Ben, I'm going to turn it over to you and ask how you think we should use the remaining time, or if people want to go, they certainly can.
Yeah, so usually what we do toward the end is if there's open questions in the Q&A that you have some perspective on, just to kind of do your best to answer them out loud. If you're looking at that right now, looks like there's a couple that are open there. Oh, you're muted.
Sorry about that. So let me share the questions. Oops. Okay, can people see the questions document? Great. So let's go to the first one. Looks like many of these are answered, but I'll go through them regardless. How to check the quality of a tree once it's prepared. So this answer is excellent. Bootstrap values are when you take the multiple sequence alignment and you build a tree for a subset of the columns in the alignment that are picked at random, and you can rebuild that tree many, many times. Typically 100 times is pretty standard. And then you can ask how often in that subsampling of the multiple sequence alignment do you wind up with the same topology? And that is notion is called a bootstrap value. So if every single of those, one of those subsamplings produces the same topology for a given split in that tree, you get a bootstrap value of 100. If 90% of the time you get that same split, that bootstrap is 90, and so on. You know, depending on how strict you are, you may or may not trust something that's at 75. But if it's at 95, then it's pretty strong. So that bootstrap value is sort of how much you should trust it. The correct answer is trust nothing. But eventually you have to take a leap of faith occasionally. And so you can, you know, at least have a confidence associated with the result there. And you could potentially try another tree building method and see if you get the same topology. Branch lengths are a tricky beast and don't really have bootstrap values in the same way as node splits. So what you really should have for a branch length, I mean, it's a dimensional reduction, right? A phylogenetic reconstruction is a dimensional reduction of the distance, pairwise distances between all of these things. And so the branch length itself represents an approximation to those distances of that that portion of the tree against everything else. But like I said, it's a dimensional reduction, so you don't. Anyway, it's bizarre. I'll stop there before I get too deep in it. Okay, is there a rule of thumb for phylogenetic tree preparation? You know, confidence. Well, you want high bootstrap values. If you're getting trees that don't have high bootstrap values, you might look at which branches are the worst participants, the greatest offenders, anyway, and leave them out so the remaining tree is more reliable. There are, you can change thresholds. Like I mentioned, G blocks is meant to reduce some of the uncertainty from hypervariable regions, and so you might be stricter with your G blocks. You know, KBase is meant to help it make it easier to play around with these things. So, and you can, you know, try changing the parameters around and see which gives you better results or results that are more confident. And the nice thing about KBase, and part of the point, is that you can then see exactly what your parameterization was, and it's easy to go back and look at how you did things and share with other people, and you have that self-transparency on what you did to wind up with the result you got. Is it possible to increase the values on nodes by increasing bootstrap during calculation? So yes, so you can change how many bootstrap runs you do. But yes, this answer is just right on the nose. If you can run longer, you can do more bootstraps, so you get more sampling and therefore greater reliability in the value that you have. I have whole genome sequence for different species. Can I construct a phylogenetic tree using both genes? Well, first of all, a tree requires 3 elements or more, or else it's just a distance. So, oh, sorry, this is saying gene families. Okay, so yes, there's concatenation. There's a utility for concatenating gene feature sets, genes from feature sets. It has the requirement that you have only a single copy of that gene family in each genome, because otherwise it doesn't know which one to pick. This is why species tree construction, as done in KBase, where you're using gene family phylogenetic markers, use single copy near universal or universal gene families, these housekeeping genes I was talking about, because that way you know that you only have the one copy and it becomes phylogenetically informative. Usually, the reason you have only one copy is because these are such essential genes that two copies would allow there to be a second one that would start to diverge and disrupt basic functioning in the organism, and therefore the paralogous duplications become toxic and don't fix in the lineage. Okay, I'm not sure if that was exactly the answer you were looking for. Sorry. I don't see an announcement of microbiome community analysis. Yes, we'll share soon. Can I change fonts or size in the tree? Well, the PNG is a bitmap image. The PDF is vector. And so if you have a vector image editing program like Adobe Illustrator or many others, you can go in and edit the vector version of it. So you can download the PDF version and edit to your heart's content, change colors, fonts, whatever. I wonder if I can type in this. Question, is there a predefined database of genomes to compare your query sequence to? Excellent question. Currently, the best route is to focus your search on related genomes, and so that's why I started with the insert genome into species tree type apps. If you have your own genomes that you've uploaded, you can certainly use those. We don't. There's functionality coming online to support whole system, not just RefSeq, gene homology search. But for right now, for the purposes of this tutorial, where you're trying to get into phylogenetic reconstruction and so on, it's best to start with a collection of related genomes using a species tree search. If you use species tree on a self-annotated with RAST, for example, genome, will the tool find relatives to this genome? Yes, absolutely. Does it use all NCBI genomes that are online to compare with? So we take non-redundant RefSeq genomes and put them in our reference database. Does this work well for viruses? Does it have a lot of viral genomes? No, not at this time. We do have a collection of tooling for, well, we have some tooling for viruses, VerSorter being the primary one. So that's on system. We aren't yet, or currently, I should say, hosting a collection of viral genomes. That may change for obvious reasons, but right now we are focused on DOE systems like soil. But yeah, that's a tricky one. I think probably you can certainly upload your own. Let me say that. Can you specify more distant genomes that is define a genome set based on a selection of relatives you're interested rather than solely that are more closely related? Yes. I mean, you could use the build microbial species tree with the phylum skeleton. That would give you a phylogenetically diverse collection, and furthermore, those were picked as sort of canonical exemplars wherever possible. Some of these lineages are only represented by mags, so you know that's what you get for those phyla. But that's one way to get a collection of more distal exemplars to play with. How good is the database for protein blast? So as I mentioned, you pick your target collection of genomes to search against. So you have to make that selection first. Is it possible to use the same analysis for fungal? Right, so everything that I've mentioned, I did all of this analysis using microbes as the examples. Some issues like functional annotation in particular are dependent on the history of the scientific research that produces those functional annotations and understanding of those gene families. That's less well, fungi are just less well characterized than bacteria for a variety of reasons. So all the sequence homology stuff, all the that sort of thing, yes. Anything based on functional annotation needs to be taken more with a grain of salt. Obviously, fungi are very important, and we're trying to expand based on whatever knowledge is extant and bring that into KBase. But please work on fungi and viruses and expand our knowledge there as for all of us. Can I use BLAST-N to do ribosomal RNA removal? We have incorporated, we in collaboration with JGI have incorporated the JGI read QC pipeline. And so filtering of various types of sequence are possible through that tool. It's the RFQC filter, I believe. Can you build a feature set of 2 genes combined? Align 2 or more genes. So you can build feature sets of non-homologous genes. So the sequence homology, you're not planning on building a gene tree of them. It depends on how you're going to use it. Typically, it's cleaner to just build feature sets of sequence homologous genes. And then if you need more than one, have more than one feature set. That would be my recommendation. I think that's the spirit of the question. Can you download a real Genbank file from KBase? Yes, there's imports, direct imports app that allow you to specify something that's not in your staging area, such as from NCBI or Genbank. And there are a few other places. Yes, and this is the answer. The type is different, or the extension of the type. Species tree method for bacteriophage phylogeny. No, I would not recommend the species tree method as implemented here. These insert species tree are based on gene families that are prevalent and housekeeping in microbes. And that's not going to work for bacteriophage. What I would recommend as an approach, and it's something I hope to have available soon, is but you could do manually before. There's just a one-stop shopping app for it. The rough idea I would implement is to take that collection of phage and do an ortho-MCL to figure out the sequence homology across that collection, determine the core complement of genes. So again, using that Pane genome slicing, and you could use the circle plot to do that slice to get the core set. Chances are they're single copy because these things are so tiny, but you could do an extra check to make sure it's a single copy representative. I doubt it's necessary for phage. And then concatenate those genes into a do MSAs, concatenate the MSAs, and then build the species tree of that concatenated MSA. That's essentially what's happening behind the scenes for the microbes, but it's using the phylogenetic markers that were predetermined for microbes as opposed to an on-the-fly Pane genome calculation. It boils down to the same process. It's just manually implemented. I hope to have a Pane genome to species tree thing on the soon side of things. It's absolutely something you'd want for viruses. And so hopefully that will be available soon as a single app, but in the meantime, you could implement it manually with these steps. So again, it would be ortho-MCL, muscle on each of the feature sets that, ortho-MCL, then Pane genome circle plot to get the core, the feature set of core. Split those up by homolog family, build an MSA for each of those homolog families, concatenate those together, and run a species tree generator like a tree generator like FastTree. And hopefully that will be available as a single app soon, but you could do it manually yourself. Could you use RAST for homocytes? Yeah, again, it's the fungal issue of annotation. There is a fungal specific app. And use the contact us infrastructure because we do have some experts who would be, I'm sure, happy to talk to you. RQC filter, that's the read quality control filter, is what that stands for. These are, yeah, you should go to BBMap to understand the options for that particular filter. I see we're over time, but so I apologize for anyone who has to go, but I'll see if we can't just finish these up anyway. What about proteins that are called, but do not have defined cog, seed, PFAM, or KZ hits? Conserved hypothetical proteins with poor functional annotation. Yeah, you can still add them to feature sets and use sequence homology. Building heat map. Oh, okay. Apparently I already answered it. Okay. This is a very interesting case, right? So some of the more interesting genes are the ones that don't have functional annotations that have been known forever. And so looking at genome context and conserved genome context can be one route to answering it. There are other guilt by association measures like expression levels, promoter regions. There's a whole school of systems biology devoted to trying to understand system components and expression and genome context and so on. And KBase is building a knowledge engine to help answer questions like this, exactly like this. So stay tuned. Can we get this to Sulfo-Pertinax tutorial as an example template? Yes, it's a public tutorial. Oh, you know what? This one has not been labeled a tutorial yet. It's public, but I don't consider it in a final enough state to call it the official tutorial yet. That will be coming soon. But you can certainly use it right now as an example template. It's public and just use the narrative ID number, or you can search for Phylogenomics tutorial. It's not an official tutorial. Fungal specific app model building, build fungal model, answered. Pane genomes, don't you have to annotate first? You need gene calls. It's going to use the sequence and not the functional annotation to build the homolog clusters. So technically, for a Pane genome, you just need the gene calls to be in place, not the functional annotation. Anyway, you get both by running raster proca. Clustered all around, origin of replication at the start. Well, so the desulfur rudis that I used as the base genome, that chromosome was defined as starting at the origin of replication. So in this particular case, because I used desulfur rudis, the beginning, and it is a single contig, it's circular, we split it, or we started the numbering for the chromosome at the origin of replication. But you're not necessarily going to get that. Depends on the base genome you pick. Compare pangenome outputs. Which one is better? Ooh, great question. It's if you want, there's no N50 equivalent single number that you can boil all this down to. Maybe there should be, but if you have more clusters with fewer members, then that would mean you are getting tighter clustering and potentially more reliable, if that's what you're going for in terms of tighter sequence families. If you want a broader picture and you are willing to, it's the classic sensitivity versus specificity trade-off, right? You can have fewer clusters with more members, or more clusters with fewer members, and it's sensitivity versus specificity. It's, you have to pick what you're going for the type of analysis you're doing, I guess I would say. Insert a genome assembly. So yes, it's you need the gene calls. An assembly doesn't have the gene calls. The genome has the gene calls. However, this particular version of the species tree is using microbial phylogenetic markers, some of which are probably just fine for using for the fungi, but take it with a grain of salt. Ribosomal RNA database, species-specific RNA sequences to remove with RQC filter. I am not an RQC filter expert, so I can't answer that. Maybe. That's no help. Any plans to run an in-person workshop on this topic? Talk to Ben.
Yeah, once people are doing in-person things again, we'll start to think about this. I mean, usually, so some of these workshops or these webinars were converted from workshops that we do at the JGI Genomics of Energy and Environment conference that happens every year, but was postponed and canceled this year. So we will be there. That's if you want to attend a lot of KBase workshops. That's one of the places that we have 2 days worth of workshops each year. So just pay attention to announcements about that. But and we are interested in doing workshops at universities, of course. So you can write to engage at kbase.us and just ask about doing a workshop at your university.
Yeah, and in person is to be determined. So this last one, sometimes recent publications, identifying new genome features. Absolutely, we're never done. Currently, we do not have the ability to edit a genome object in place to add features. I would recommend to do that offline, upload it to your staging area, and import. You just use either Genbank's sort of hard to generate manually, but GFF is super easy to make. So you could take the assembly sequence as a FASTA and the gene coordinates as a GFF, and then upload those both to your edit the GFF, upload those both to your staging area, and then import the GFF plus FASTA as a genome object from your staging area, and then you can dive into all this tooling with that object. But yeah, you sort of have to do it manually online at this time. Okay, so that's all I'm seeing. And I guess I'll hand it back to Ben to close things out. Thank you all so much. I always enjoy sharing this with people, and these days especially, I think webinars are a good way to go. But you can always use the contact us infrastructure to ask more questions or dive into KBase and please, we always want feedback because it helps us make KBase better for you. And we learn a lot from it. So thanks, everyone.
Thanks,
Ben,
Dylan. Yeah, thanks again for this great presentation, and especially for answering all those questions at the end. Like Dylan said, we love feedback. So if you go to the top of the Q&A document, there's a feedback form. As soon as the recording is done rendering on my computer, I'll send it around to everybody. Thanks again, everybody, for attending, and we look forward to having you out for more of these webinars. So just a reminder, we've got one coming up on May 20th. We'll send out an announcement for that soon. And take care, everyone.
Thanks for watching this webinar. For more webinars and tutorials, subscribe to our YouTube channel. You can see announcements for upcoming webinars and recordings for previous webinars on our website at kbase.us/learn. Let us know in the comments what content you'd like to see in future webinars. Be sure to follow us on Twitter at DOE-KBase. And if you have questions or encounter an error when using KBase, please contact the help desk.